All right. So it helps me a little bit to get a sense for the audience. So I want to start with a couple of questions. Is anyone in the room, and you can do this by a show of hands, currently suffering from what would be called a chronic complex disease? These are things like heart disease, hypertension, diabetes, Alzheimer's, obesity, arthritis, Crohn's disease, chronic kidney disease, depression. Okay, a lot of you didn't raise your hands, which means I know some of you are fibbing. Because 76% of US adults suffer from at least one chronic health problem, and about 50% from at least two. That's a lot. Wouldn't it be great if we had a way to not only monitor your risk for these chronic diseases, but also to sort of help you pick the right ways to either delay their onset or to adjust when you finally do get them? That's what we'd like to talk about this evening. I have a related second question. Maybe we'll get some more hands, maybe we'll get fewer. How many of you have ever had your genome sequenced? Oh, great. I think we got more there than we got for chronic disease. Right? Okay, so your genome is the book. This is all the DNA you have in all of your cells. This is the thing that defines you, describes who you are, describes how you respond to things. And in today's day and age, you can go and pay a company to have your genome sequenced. So there was, a handful or a smattering of folks that raised their hand. Why did you have your genome sequenced? Was it because you were worried about finding out whether you might have one of these diseases? So at least one or two. That's not generally the reason we get our genome sequenced. We generally do because we want to find out if we have an old lost uncle somewhere in Nebraska, right? It's not because we think it's going to help us with disease. Worse yet, doctors know this. So I have yet to find anyone whose doctor has said, you know what would help here? Let's have you sequenced. Not usually what happens. What happens when you do go to the doctor's office? What are the 3 metrics that doctors take every time you walk in? They weigh you. I don't like that one. They take your blood pressure. Mary doesn't like that one. And they take your temp. Right? They don't necessarily do anything else routinely when you walk in, other than maybe talk to you. But then these days, have you been to the doctor lately? Then they're like 10 minutes. They don't even really talk to you anymore. Okay? What I want, what would be a fantastic goal, would be to have a way to take a sample, let's say it's a blood sample, and then from something in that blood sample, give me back what's my risk for these chronic, consistent diseases. Wouldn't that be great? Would that change the way you behaved? It might. For a lot of people, it wouldn't. Absolutely. I actually ask this question occasionally in the class I teach, and a lot of them are like, I don't want to know. Ignorance is bliss. But maybe you do. Now, some of you have to be sitting there going, wait a minute. Don't we already do this? Don't you walk into the doctor's office and sometimes they take a blood test? But they don't do it every time you go, right? And usually when you go, if they take a blood test, they'll ask for a CBC. A CBC measures more or less all the things that tell you whether you have an infection, or the levels of various blood cell types. It's measuring proteins mostly. There are separate tests they can do, like a lipid panel to measure your cholesterol levels. But at least in my experience, when a doctor orders a blood test, they already suspect something. They're not doing this to monitor you, but maybe to diagnose something or to give them a hint. So it's not really giving you a, hey, how likely are you to have heart disease? Or, hey, how likely are you to have diabetes? So what I want to tell you today is that, yes, I have, okay, the pointer works. I have another pointer here too. Is that when you go into the doctor's office, they will measure things like proteins, And you might get your genome sequenced because you want to find out about that late uncle in Nebraska. But the central dogma says that the DNA makes RNA, makes protein. And we have now, for years, said RNA is just a messenger. It takes things from the DNA and walks them out here to turn them into the protein. And what I want to convince you of tonight is that that is an oversimplification of RNA, and that RNA actually gives you a lot of information about your health, and more specifically, that there are certain levels that your RNA should be in for you to be healthy. And when it gets too high or it gets too low, it's like Goldilocks with the bowls of porridge. That's not good. All right. So I told you that you don't routinely go to the doctor's office and get your blood drawn, right? I turn out to be an exception to this. I go to the doctor every year, and the first thing they do is they take my blood. And they take my blood because I have sugar problems. More specifically, they want to know how I've done this last year in managing my sugar problems. I would like to know more often than once a year. So I actually get up every morning and take my blood sugar first thing in the morning. And I live perennially right at this line between the yellow and the orange zone. I am technically diagnosed as a diabetic. And it stinks, okay? If I eat white rice, I go into this orange zone. If I am starting to come down with a cold, I actually know it because my blood sugar will creep into that orange zone long before I have any symptoms. And God forbid I should ever eat a cookie. Okay? That's diabetes for you. Your system cannot manage blood sugar in a normal way. Now here's the kicker. I am also reactive hypoglycemic. So if I drink alcohol, I go to the blue zone. If I exercise too aggressively, maybe you can tell, I go to the blue zone. If I ride a roller coaster, I will often go so far into the blue zone that I pass out. That's an adrenaline response. That's a rare reaction. Being both, I'm just blessed. Okay? And I come by this entirely naturally. I was gestationally diabetic with my second son in the front row here. My father is a type 2 diabetic. Every single extended family member on my dad's side over the age of 55 is a diagnosed diabetic. My mother's side has problems with hypoglycemia. Yay me! Okay? And I worry about both of my kids and what they're going to get in terms of blood sugar levels. The reason I'm talking about my family is that tells you that some aspect of my diabetes is inherited. It is genetic. It comes from that genome that I could have sequenced, right? And luckily, with modern tools, we can do things like ask what genes in the genome associate with diabetes. And our technologies have been getting better and better, and we started out with a few genes, and then in 2014, we started looking whole genome-wide because we could now sequence them pretty well. And we're up to at least 700 different genes in the genome that influence whether or not you're going to become a diabetic. That's an enormous number. Honestly, it makes me question whether it gives me any more information than just knowing my dad has it. So I haven't been genome sequenced either. And it only explains about 30% of type 2 diabetic risk. So yes, we know that diabetes is often genetic, but the genes by themselves are not the whole story. This turns out to be true for lots and lots of things, but that doesn't stop us from spending lots of money to sequence lots of people. And we sequence folks with a disease, and we sequence folks without a disease, and then we say, how do they differ? What is the positions in the genome that are different in those with the disease versus those without? And from that, we've been able to explain an enormous amount of what I would call rare genetic disease. These are things like kids that are born with something that's really terrible. But in common disease, the types of things that you and I would get in our a little bit older age, we're actually doing pretty poor. Don't get me wrong. We have lots and lots of genes, 700 for diabetes alone. But it doesn't explain everybody that comes down with it. So why is that? Well, that's because the genes aren't the whole story. Right? There's lots of other things that contribute to risk for whatever your favorite chronic disease is, if anybody has a favorite, right? My blood sugar issues mean I'm thirsty all the time. All right. I have to admit that when I look at all of these things that contribute to whether or not you'll become a type 2 diabetic, I take a pause at this one down here. And I think, does my depression contribute to my type 2, or am I just depressed because I can't eat that cookie? Right? So to this point, what you should be thinking to yourself is that you are more, wrong direction. You're more than just your genome. Just like a book, a story, or if we had a communication, it's more than just the words on the printed page. You guys didn't come here to read my talk, right? You came here to hear it, because we communicate better through spoken language. Because in spoken language, we can have emphasis. And emphasis can, in some ways, change the meaning of something. For example, I don't think he should get that job. 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 And this is why, if you're arguing, don't do it by text. Okay? Listening, communicating, emphasis can change the meaning in subtle ways. The genome's the book, it's the sentence. But what's the sound? What do we hear out of a cell? That, my friends, is the RNA. The RNA is the genome speaking to us. And it provides emphasis, and it provides color, and it provides interest, at least for me. Okay? So what I'm going to tell you now is that we're going to go through how we measure RNA, and I'm going to show you through a series of simple examples that the RNA not only reflects your genes, your DNA, but also the environment and all the things that you've been exposed to and done in your life. Some of you may not want to know what you've done in your life. Right? All right, so I can take any random person. We're going to start with this little cute guy. And I can take cells out of that individual. And within each cell, and there are literally billions of these, and within each cell, I have my genome. And my genome comes in 23 copies of chromosomes, 2 of each, one from mom, one from dad. And on each one of those chromosomes, you have some number of genes. That's my little red ticks here, because I'm not a good artist. Okay? And from each one of those genes, that gene is singing to you by producing some amount of RNA. 2 copies in the DNA, but potentially many copies of RNA, depending on where that emphasis is. Okay? This is no different than taking that reference book and taking it to a copier, and you make as many pieces of the same page as you would like. So I can make lots and lots of copies. And luckily, I can measure those copies very, very easily these days with something called RNA-seq. And with RNA-seq, I can get an absolute measure of abundance on any gene's RNA production levels. And when I do this for an individual, I see that across the genome, we have about 20,000 genes. Different genes are at different levels. Okay? So RNA varies by which gene we're talking about. So if I am measuring RNA to learn something about the individual, I have to tackle a couple of problems. One is, what tissue do I start with? So it turns out almost every cell in your body carries the same genome. It's part of what makes getting your DNA easy, getting your genome sequence relatively easy. Pop quiz, which one doesn't? Red blood cells do not. But They all carry the same reference book, but you have bone cells, you have brain cells, you have heart cells, you have little pinky toe cells, right? What makes them different from each other? They look different, they do different things. It's because they're reading different parts of the book. They're playing different notes of the music, if you want to think about it that way. So if I take one person again, and take different tissues, blood, skin, liver, for the same gene, I will get different levels. Okay, so I can distinguish, actually, which tissue I have by the fingerprint of which genes are at what levels. What about twins? Okay, my lab is sitting up there in the upper left-hand corner. They're going to love this. Twins look very, very similar when they're young. And as they age, they diverge. And the older they get, the less they look like each other. Because they exercise at different amounts, they're exposed to different medications, they eat different things, they might get exposed to different pollutants, and all kinds of other things in the environment, until you get to this picture, where one of these two twins is graduating from my lab tomorrow with her PhD, and the other one doesn't even like science. She's studying to be a therapist. They diverge. Does the RNA reflect this? Actually, yes. RNA, when I pull it from two identical twins, doesn't look identical. How about if I take the RNA and I do one of these perturbations? I can take an individual and look at them before and after a drug, and those genes, again, are going to move around. So I have a measurement now that can get to me variation in the genes, variation in the tissues, variation in the environment, any of those environments, stimulations. And so RNA turns out to be like a handle for measuring variation within the population. Okay, now some of you are probably thinking, oh, if everything varies, how are you going to get anything out of that? And I have had days I've thought that. Okay? But what I want to talk to you the rest of the day about is not the variation in these things, but the variation between you and the person sitting to your left or your right. Because if I take a handful of people and I look at the same gene and I measure those RNA levels, they're going to look different. And the question I want to pose today is, do those differences tell me anything about their disease state? So we're going to look at real data now. Don't worry, it'll be okay. Okay? And we're going to do this with an NIH-funded project called the Gene Tissue Expression Project, or GTEx is what we always refer to it in the lab. And in GTEx, they sequenced RNA from over 800 people from an assortment of tissues. There's got to be someone here wondering, wow, they got the tissues. And that's okay. It's a broad variety of folks. And we want to ask questions about, like, for example, if I look at a single gene, I told you it varies from person to person, how much does it actually vary? So I can take all 800 people, line them up, look at one gene, sort it from highest expressing most amount of RNA for this gene to lowest expression of this gene, and I tend to get curves that look like this. What changes is the absolute number over here. Sometimes they're relatively small, sometimes they're quite large. We're going to pause here for just a second and make sure everybody can understand my weird data diagrams, okay? I'm lining people up here by expression levels. I can instead, for any expression level, count how many people actually have that expression level and produce a histogram like this. And from that, you can see that most people's expression of this gene is pretty low, and there's a few people that are screamingly higher. And for the sake of space on a page, I can take this histogram, rotate it one more time, and get a violin plot, where it's fat down here because, again, most people are lowly expressed, and it's got a few people up here because those are the extremes or the outliers. And if all of that is way too much math for you, that's okay. All you have to remember is that in the black box is 50% of the population. Okay? So remember I said GTEx did this for 800 people across 30 tissues. This happens to be one form of adipose tissue for one gene, RCAN1, this guy right here. And now I can look at that same gene across many tissues, and as I told you earlier in the cartoons, it jumps all over the place. This gene is lowly expressed in adrenal glands. And in those 30 tissues, and oh boy, there's a bunch of them, it starts to look overwhelming. But some people, you can see that dot way up there, are screaming for this gene. It's like the music or the spoken language is yelling. I want to be able to quantify this because I want to be able to understand when a gene is high versus when a gene is too low doesn't matter. So I'm going to quantify this in, admittedly, a really simple way. I'm going to take those people, I'm going to line them up, but now I'm going to line them up by the expression level of one particular gene. The person who's the highest is over here, the person who's the lowest over there. That first peach-colored plot I showed you did exactly that. Okay? Now I'm going to take the highest person, maybe that's you. And the lowest person, maybe that's me. And I'm going to calculate the ratio of those two people and say there's a 100-fold difference in the expression of this gene across the population. Now notice that that kind of ignores the fact that my husband, who is the most normal person you will ever meet, and he's over there, and turning red, is sitting right here in the middle. But it gives you a sense for the range. Okay? So now I can take blood, which is by far the easiest tissue to get for any population, and I can take every gene in the genome, and I can calculate that one number for every gene. And as you can see, some of them have very little variation across the population. The average is actually surprisingly high. And I have a very long tail. Realize there are genes where the maximum and the minimum person have 10,000-fold difference in how much RNA they're producing. That's enormous. I got worried about that. I started thinking, because I wear my statistician hat sometimes, or my CS hat sometimes, or my biology hat sometimes, and I started thinking, what if this is being dominated by outliers, and what if the outliers aren't real? Like, they're real people, they're real samples, but maybe something happened along the way. Okay? So then, what we decided to do was to throw them away. Okay? So we then take the bottom 10%, the top 10%, and we ignore them, and we recalculate our ratio, say between Roy, wherever he went, sitting over there, and Amy, who I don't think is here today, but that's okay. And now we get this trimmed mean, and we want to know what that distribution is. In other words, how many of the people, or how much of this huge variation is just being driven by these people at the extremes? And that distribution looks like this. And on average, it's almost 6-fold between the min and the max expression level in any two random people. That's quite a bit. This is in blood. If I look at a different tissue, like brain, I hope somebody's thinking, how did they get brain samples from 800 people? It shifts. Your brain is actually far less variable than blood. And the average there is only about 3-fold. Turns out for brain, we only have about 100 people. One of the unfortunate off-color jokes about GTEx is this is the crash test dummy database. Almost all of these samples are from people who were in car accidents or on life support for some other reason. Gruesome a little bit, but gives us a lot of data. All right, so what I've told you so far is that humans are highly variable. Some of you are going, well, no duh, I can look around and see that. But I was surprised at the extent of the variation within the RNA. But I was also actually quite disappointed because the NIH spent millions of dollars on the GTEx data set, and all we got was RNA. And the question I want to ask is what comes with high RNA, what comes with low RNA. Is there ranges that the RNA is okay? And we don't have the medical records with GTEx to do that. That's kind of a bummer. Okay? But right here in Colorado, there's an alternative data set where we have this information. This is actually kind of a rarity. This is data that is being generated by the Global Down Syndrome Foundation down in Denver, where they are taking samples from individuals with Down syndrome and unaffected, often age or gender matched individuals. And then they're doing not only genomic sequencing, but RNA sequencing, and I have lots and lots and lots of clinical data, which is pretty cool. And it just so happens that about 3/4 of these people have Down syndrome. So what is Down syndrome? Down syndrome is when you are born with an extra copy of chromosome 21. Remember I said we get 2 copies, one from each parent, of all 23 chromosomes? In the case of Down syndrome, a mistake was made, and you got an extra one. That's less than 1% of your genome. So they have a little bit extra. With Down syndrome, they have very characteristic facial morphologies, certain intellectual disabilities, and more importantly, their prevalence of all these different diseases has shifted. So if you have Down syndrome, you will more or less get Alzheimer's by sometime in your 40s. They have an increased incidence of autism, epilepsy, obstructive sleep apnea, but remarkably, they almost never get solid tumor cancers. So they're like Supermen in that regard. So what is it about that little bit of extra DNA that gives rise to this altered prevalence of all these different disorders? And we, for a number of reasons, suspected it might be in that RNA. So what we absolutely know is true is they have 3 copies of chromosome 21, and that RNA levels roughly follow DNA levels. So if I have 2 copies and I make this much RNA, at 3 copies I will make 50% more, or 1.5-fold, 1.5x. Okay, now stop for a second and remember that a few slides ago I said that the max and the min person were 2 to 6-fold different in their expression levels. 1.5? That's little. Like, part of me wonders why they have any problems at all. But they do. So what do I expect an individual with Down syndrome's RNA to look like? So here is their 100 people without Down syndrome, what they affectionately refer to as typicals. Okay? Here's the distribution of one particular gene. I've actually been showing you the same gene all night. Okay? Here's the distribution of expression levels when you have 2 copies. If I take one person out of this distribution, multiply it by 1.5, in other words, give them 50% more, I expect the dot to be showing up here. And I can do this for every person in that distribution and get what I expect a 1.5-fold increase to be. And then I compare that to what I actually see. And actually, they're strikingly the same. And we had an entire paper on the fact that they don't change. Like, the 1.5 seems to be solid. But what I also want you to notice is that when you look at these 2 distributions side by side, a large fraction of the people in the red zone, the trisomy individuals, the new RNA levels that they have after having trisomy, they fit in the same distribution. They're in the same range as somebody without trisomy. This led us to wonder if the problem was not actually that they got an extra chromosome per se, but rather that sometimes that extra chromosome drives you outside of typical range. That typicals could define a healthy typical range of RNA levels, and that maybe when you're outside of that, that's when they're affected by those comorbidities. So we wanted to ask this, and was like, okay, I'm wearing my statistician hat, my genetics hat, and I'm like, okay, we just need to know what is the normal healthy range for any given gene. So we have some genes like this one that are transcribed anywhere from 0 to 10,000 copies. And we have other genes like this one, where the variation between the lowest and the highest is much, much smaller, and in most people are within about 500, a zone of about 500 of each other. Which means sometimes the typical normal healthy range is really broad, and sometimes the typical normal healthy range is really narrow. So I've got to do this by gene, can't do some generic cutoffs. And if I put my statistician hat on, I could say, oh, where are my outliers, and do all those things, and you guys would all fall asleep because it's math. The other problem is that presupposes that all of my typical individuals are healthy. And we started out by noting that 75% of people suffer from some kind of a chronic health discrepancy. I don't think I can make the assumption that all of the people in my non-trisomy set are normal and healthy. And yes, I can't make assumptions about they're healthy. I probably can't make assumptions about whether they're normal either. I'm certainly not normal. Now, we often know what's not normal, but it's harder to say what is normal. So rather than try to, and admittedly, Mary and I spent more time than I'd like to admit on these two slides trying to figure out what normal and healthy actually means before we gave up. And we punted. And we said, can we do this a different way? Do we have to make that definition? Okay? It turns out we don't. We can go back to this idea of sorting people by rank order. Let's put the high ones over here and the low ones over here for our favorite gene. And what we want to get out of this ranking somehow is, does the expression level of this gene in some way associate with a particular disease? And we built an algorithm for this because at heart I'm still a computer scientist. And we call this attribute rank enrichment analysis, or just area for short. Okay? And the concept behind area is that if I'm extreme, either extremely too high or extremely too low, and that extremeness somehow associates with disease, all the disease people should shuffle to one end or the other. Imagine for the moment that you go to my son's high school, and you line up everybody in the high school by height. Where do you think the basketball players will end up? Right? Right here. Is everyone that's tall going to be on the basketball team? Is everybody on the basketball team going to be tall? No, we're looking for trends, okay? So if everything is random, and now I'm going to use what I call the dreaded purple head disease, okay? And if the disease distribution in my population is totally random, then I'm going to follow this expected trend line basically as to where I expect to see these folks. And I can quantify this. I can start on the left and traverse one person after another, and every time I hit somebody with my purple head disease, I get to take a step up. And if there is no association between my gene and the disease, this is going to look random. The green line is going to kind of follow the dashed line, and the area under the curve between those two is going to be pretty small. But if it's like the basketball team, or in this case, my favorite gene, they're going to shift to the left. It's going to climb faster, and now I have a big chunk of area. Oh, it was a pun. And my area score is going to get bigger. Okay? So now we can take the Human Trisomy Project, all the genes in the human genome, and every disease that we have anyone afflicted with in the database. Well, we need more than a few people to have any really statistical signature. And ask for a given gene like RCAN, which I've been showing you guys all evening, what diseases associate with it. So remember, this is our typical individuals, disomies, not trisomies. These are our trisomies. We see this shift up. And one of the things that scores really, really highly for RCAN is this thing called obstructive sleep apnea. So what I've done now is I've taken all the individuals with Down syndrome and split them out into two groups, those that have the diagnosis and those that don't. But remember, I said all the basketball players aren't going to end up over to the left. And all the people that are tall aren't going to play basketball. Area not only gives me this linkage between the gene and the disease, but tells me the expression level for that gene at which the risk for that disease changes. And so if you're above this level, your odds of having obstructive sleep apnea are much, much higher than if you're below it. And in fact, if I split the population on that level, if RCAN is too high, 42% of those individuals end up with obstructive sleep apnea. If RCAN is within the disomic range, below that level, only about 16%. So now I can associate genes with particular diseases and give you a risk calculation. So, sounds cool, right? Is it real? I have a pen here that actually says, nice story, but show me the data. Okay? So, in this particular instance, we have a collaborator, Charles Hoffer, over at the Institute of Behavioral Genetics on campus, one of my many collaborators. He has an RCAN, or he has a mouse model of trisomy. So it has 3 copies of the similar places to chromosome 21, and it has 3 copies of RCAN. And they molecularly manipulated one of those copies to break it. So that the mouse now still has 3 copies, but in that one spot is broken. So for RCAN only, it has the same dose as you and me. And those mice sleep completely normally, which is pretty cool. Okay, that's a lot. I'm going to pause here, but I'm going to pause here because I'm a teacher with a quiz. Now it's quiz time. You didn't know there was quiz time, didn't you? Okay. Remember I said, well, I can look across all these genes, and some of them are very, very broad, and some of them are very, very narrow. And when I'm an individual with Down syndrome, and that extra 50% pops me up, which genes are more likely to be associated with disease? Give you a second to think about it. Then we're going to do this by show of hands, because I know you all know how to raise your hands. We've done that part before. Okay. How many of you think they're going to be with the gene A group? And how many of you think it's going to be the gene B? Oh, y'all are so good. All right. So when we looked at all the genes, we linked to all the diseases for Down syndrome, there was this really cool pattern. When the typical range was small, narrow, the amount of people we would expect to climb out of that range would be most people with Down syndrome. And in fact, if NAR2 is one of those, and those were the disease associations that more or less everyone with Down syndrome has. Low IQ, facial morphologies, interferonopathy, so they don't respond to infection the same way. When the typical people have this broad range, fewer people could get kicked, only those that are close already to the edge would get kicked outside of range. That would affect many, many, many fewer people. And those were the comorbidities that were elevated in Down syndrome, but didn't affect everyone with Down syndrome. And in fact, to some extent, the width of this distribution in the typicals could predict for us how much more the elevation was in Down syndrome. That's quite shocking. That suggests that it really is just that the RNA levels are getting too high, and that's pushing these individuals into disease. All right, so we've talked about being too high, but I started by saying this was a Goldilocks talk, which means we got to talk a little bit about whether or not it matters to get too low. Okay? So, I hate to break it to you, but everyone in this room is a mutant. Okay? The average human has 100 genes in your genome where you only have one functional copy. Either your mom or your dad gave you a broken one. You can decide who to blame later. Okay? And when that one copy is broken, you get half as much, often half as much RNA, sometimes just half as much protein, but usually half as much RNA. Matter? I mean, we're all sitting here. We're all okay. Wait a minute. Some of you raised your hands earlier when we talked about chronic disease. Same quiz, different scenario. If I lose one copy, are those going to be a problem? Are those going to show up more in genes that are broad or genes that are narrow? So it's important how I phrase this question. Are you going to have them in the genes that are broad, or are you going to have them in the genes that are narrow? I'm not asking where they're a problem, I'm asking where they're going to show up. Okay? So how many of you think it's in the broad range? Okay. How many of you think it's in the narrow genes? Okay. You weren't quite as confident on this one, and that's okay. Turns out that when you look across a very large population of people, almost all loss-of-function mutations show up in these broad genes. Genes that vary a lot in the population. Because think about it. If I'm going to lose 50%, I'm much more likely to be okay to lose 50% somewhere in here than here. This is a classic example of what we call in statistics survivorship bias. Okay, don't think I'm crazy because now there's an airplane on the tape. But the classic story about survivorship bias comes from World War II. Airplanes would go out, they'd get into dogfights, they'd bomb Europe, they'd come back, and they would measure in red dots everywhere there was a bullet hole in the plane. And they were like, oh, there's lots of bullet holes in these areas. We need to put more armor there. Right? Until somebody said, no. If there's a bullet hole here, or if there's a bullet hole in one of those other spots, the plane didn't come back. Which means we should put our armor there. It's the same thing. We can't afford to lose when it's tight. Okay? How do we know this is true? Those same genes that have tight distributions where we don't see loss-of-function mutations, those are often the ones that are associated with those rare, terrible diseases that kids get when they get what I would call a milder mutation. It doesn't totally lose it, but then they still have disease. We also know this because there's a few unfortunate individuals born with a loss-of-function mutation in a tumor suppressor, and their odds of cancer go up astronomically at some point in their life. They're like already halfway down the path. Doesn't kill them, isn't good for them. So RNA levels do look like they have to stay in this Goldilocks zone. It's not good for us for them to be too high. We saw that with the Down syndrome individuals. It's not good for us to be too low. Matter of fact, we don't even see a lot of the too lows, which means we really need our porridge to be at the right temp. So I want to make one more point about the HTP before I move away from it, because I think it's a really important point, and it's actually when my lab is just starting to really go down heavy. Area and our techniques ask if one gene is associated with one disease. And it works pretty good, and we have lots of statistics and data to back it up. But we're also finding that it's almost never one gene. So this is like the toughest graph I'm going to show you, so we're going to walk through it slowly. This is for obesity. That was the disease we were looking at here. There are two genes that associate with obesity. Again, I could tell you from Area's results what level is too high. If this gene is too high, your odds of being obese are about 10%. If this gene is too high, it's about 12%. Across the entire Down syndrome population, it's about a 10% rate. If they're both low, you are much less likely to be obese. And unfortunately, if they're both high, your odds are screaming. With 400 people, we really only have statistical confidence to do pairs of genes. If we had more people and more data, we could go to 3 genes, or 4 genes, or 5 genes. And you can imagine a scenario where we could identify a fingerprint of genes that's like, hey, if you have this set of things too high, maybe you should change what you're eating. Or maybe there's some other thing we should be doing for you. All right, so I want to tell you briefly about one other data set we looked at. We got a little bit concerned about the fact that our human trisomy data set was 3/4 individuals with Down syndrome. And that really, if we're going to use this in the general public, we need to look at data sets that are JP public. And we went out and looked around, and there's one that caught our attention. And that's this one, where 324 first responders to 9/11, they went and they worked the scene, and some fraction of them, in this particular data set, 801 of them, came down at some point afterwards with PTSD. We have blood samples from all of them, all 324, and we know whether they currently have PTSD, previously had PTSD, or have never had PTSD. They were all exposed to the same stressful event, but only some of them got PTSD. So we run our area algorithm on it, and we find a number of genes that associate with PTSD. BRD4 happens to be not even the top hit, but it's up there. The reason I show you BRD4, as in a mouse model of PTSD, if you reduce the levels of BRD4, the mice no longer really show PTSD symptoms, and they don't respond to stress in the same way. They respond much more like the groups that never get PTSD. So we actually take this as a validated hit. So we took the set of hits that we get, a few hundred genes that we had, and we said, what do they have in common? We ran into this thing called gene ontology analysis, and it tells us that in the set of genes that we associate with PTSD, they're involved in things like heart and vasculature and steroid hormone receptor. And sure enough, if you go look in the literature, people with PTSD have heart problems, and they have elevated hormone signaling. And here's where I get to say buyer beware. We don't know, in the case of PTSD, for most of these genes, whether or not the RNA levels being up are causing PTSD, or pushing you towards PTSD, or whether or not because you have PTSD, the RNA levels are going up. Classical chicken or the egg. Now with many of our diseases, and this is the reason it says sometimes, we do have some knowns. Not because area told us anything, but because the literature and great scientists like Roy and Leslie do really great work. We, on the other hand, now have a whole slew of things that fall into these two categories that we've got to better understand. And I'm sorry, I wouldn't be a good statistician if I didn't also say that maybe some of them are just garbage. Okay. But even if all we get out of this are these two, these are biomarkers, right? They're telling us something about the disease state of the individual, and we can learn from these genes more about how that disease came into being, potentially how to treat it, or if you go back to the way I started the talk, what's the probability of this person getting the disease? We can kind of check that box. And when we get lucky, and we're up here, and right now we don't really know what fractions sit up there, but that's what we're working on. When we get lucky and they're causal, it suggests potential new ways to treat these diseases, right? With Arcan, maybe there's some way to knock it down and help people with obstructive sleep apnea to sleep better. With PTSD, if we can reduce BRD4 levels, maybe they'll stress less. All right, so what I've told you today is that humans are highly variable, that this variability in expression levels helps to explain why in Down syndrome some folks get these comorbidities and others don't, and that RNA levels have this Goldilocks zone where you really can't be too high or your odds of disease go up, and you really can't be too low or your odds of disease go up. And what we'd like to work ourselves to with a technique like this is a different way of thinking about medicine. So now this is my vision, right? This is where we want to go with it. We want to be able to go to the doctor and give them a sample, sticking with blood, though my niece will say needles are not nice, and take blood samples and be able to track something in that blood that says, hey, my risk of disease, I'm getting closer to disease. Maybe I should do something about that early. Maybe I shouldn't wait until I'm sick. Maybe I shouldn't wait until I'm limping to go see the doctor. Maybe I can do something about that beforehand. So, case in point, I'm going to go back to my own diabetes story. I first found out that I had problems with blood sugar, other than family history, when I was pregnant with my second son. For the next decade plus, he's now 14, I was able to manage my blood sugar by checking my blood sugar all the time and watching what I ate. And that slowed everything down, because most people who are gestationally diabetic and are subsequently going to become diabetic do so within 5 years. I actually just got my full-blown diabetes diagnosis a month ago, 14 years later. So I couldn't fix it, but I could slow it down. That's because, and the reason I think RNA is so important, is because you can change it. You can potentially modify those behaviors based on what the risk tells you, and possibly change your behavior to slow it down. Or maybe even fix it. So that's all I have to say today. I will take questions. There's several people I have to thank. That crew over there, that's my lab. They're the folks that actually do this. Chris Ozeroff, he's the one that actually wrote the area algorithm. Bella, she is an undergrad here at CU who ran a huge number of these data sets. She did all the PTSD work. She also done some stuff on neuroblastoma. She rocks. And then Natalie, in a rotation in my lab, so she was in the lab for 10 weeks, helped develop the algorithm that actually finds that cutoff to say, hey, this is when you're too high and this is when you're too low. This is mostly funded actually by the Linda Cernig Institute through the Boulder branch, but we also have money from NIH and NSF when they're still functional. Sorry, did I say that out loud? And with that, I'll take any questions.